So this is Chapter 3, Part 2. So we're moving on to external structures on the different types of bacteria. So we'll start with appendages, and we know our appendages are like our arms and legs. So the appendages on bacteria kind of stick off in the same way. So the first one we'll talk about is the different structures that allow for motility. And, you know, when we're discussing bacteria and their movement, we use the word motile or motility. So flagella and axial filaments allow for bacteria to be motile. Attachment points or channels, such as Fimbriae, Pili, and nanotubes or nanowires, also are types of appendages on bacteria. So the first one we'll talk about is flagella or flagellum. The primary function is motility, like I said. There are three distinct parts on a flagella: the filament, the hook, and the basal body. And you can see those three parts on this picture. I can't say that I won't have something on the test about this, so be aware of the kind of how the flagella is structured. So the filament is the tail-like whip. The hook is that angled, angled, what appears to be PVC pipe in this picture. This guy right here. Of course, the basal body is kind of like the part that's stuck to the bacteria. This is where the tail emerges from the bacteria itself. And you can see they kind of have shown here in the picture the outer membrane, the cell wall, and also the cytoplasmic membrane. So flagella can be arranged in different ways. Polar arrangement is when the flagella is attached, or when they say polar arrangement, it means that the flagella is attached at one or both ends of the cell. So there's monotrichous, lithotrichous, amphitrichous, and peritrichous. Monotrichous is a single flagellum out one end of the cell. Lithotrichous is small bunches or tufts of flagella emerging from the same site. So instead of just a single flagellum, it's a bunch of little flagella coming out. Amphitrichous is when there is a flagella at both poles or both ends of the cell, which you can imagine makes that cell move around in a strange way. And then peritrichous, peri being around, or the prefix. Sorry, I've lost my words. The prefix peri, meaning around. So in a peritrichous arrangement, the flagella are dispersed randomly over the surface of the cell, so all around the cell. On the outside, of course. So here's some pictures of those. You're not going to have to ID those, but it's fun to check this stuff out in real life. So as far as flagellar function, obviously the whole point of having a flagella is to get the cell moving, is to allow the cell to move. And we refer to movement of bacteria in response to specific chemical signals. We refer to that as chemotaxy or chemotaxis. Positive chemotaxis is the movement toward a favorable chemical stimulus. Just like if you're at the beach and maybe you like Thrasher's French fries, most people, when they go down to Ocean City, Maryland, they find their way to that stand because of the favorable smell of those French fries. Negative chemotaxis, however, is movement away. So, somebody, sprays an unpleasant perfume or wears too much cologne, then, we want to move away. It's a repellent. So away from that particular chemical source. So either towards it is positive, negative is away from it. When we refer to runs and tumbles, a run is basically when the flagellum is rotating counterclockwise, and that results in a smooth and linear direction of that bacteria, whereas a tumble is the reversal of the direction of the flagellum, and that causes the cell to stop and change course. And we have some images here, which, you know, you're not going to have to ID these on the test, but again, it's kind of fun to see some of this stuff. And then it kind of just shows the difference between a tumble and a run and so on. Back to that periplasmic flagella we mentioned earlier. Spirochetes have that periplasmic flagella, and these guys possess an unusual wriggly mode of locomotion caused by two or more long coiled threads, the periplasmic flagella, or axial filaments. So that periplasmic flagella is also referred to as an axial filament. So it is enclosed, that flagellum is enclosed in the space between the cell wall and the cytoplasmic membrane, as I believe I mentioned before. Other appendages, so either for attachment or for the formation of channels, or in my terms, you could say like a little bit of a bridge. So you can mentally picture that it's some appendages are bridges between the cells. So Fimbriae, or Fimbria, are small bristle-like fibers. They sprout off the surface of many bacterial cells. And I generally refer to these guys as like Velcro. They allow for tight adhesion between the fimbriae and the epithelial cells, which is vital in order for bacteria to be actually able to colonize and infect the host tissues. So if the bacteria doesn't have a firm grip on the epithelial cells, say, in your throat or in your stomach or, wherever it might be making you, sick, then it's not going to be able to infect you. So that's why it's important to keep your normal flora intact, so these guys can't get a good foothold on you and make you sick. More appendages. So as I mentioned earlier, channel formation, pili, nanotubes, pili. So these are used in conjugation between bacterial cells, or the union between bacterial cells. So the pilis are able to, I kind of say it's like the arms reaching out, it's those bridges reaching out to touch other cells. And when we talk about, the occurrence of bacteria, or any microbe per se, the ability of it to pick up information, or specifically genetic information from other cells, this is how it happens, at least in bacteria. So pilis or pili are used in conjugation between bacterial cells. The pilis, or pili, are well characterized in gram-negative bacteria. Type 4, excuse me, pilis can transfer genetic material, act like fimbriae, as in these pili can help it stick and assist with attachment. And also, these can also act like flagella and make the bacterium motile. Not all pili can do that, but specifically type 4 can. And that topic, the type 4 pilis, I think is new to this edition of the book. So that's something I had never actually heard of before. So nanotubes or nanowires are very thin, long tubular extensions of the cytoplasmic membrane, and they're used as channels to transfer amino acids or to harvest energy by shuttling electrons to iron-rich substances. So there's a picture of a couple of bacteria with some fimbriae and also some pili. So you can see when I say the fimbriae are kind of like Velcro, or even if you've ever worked with felt, like felt will stick to felt. So I think of this as like a layer of that felt. It's kind of cool stuff there. These are actual images of bacteria. So more on the different stuff on the outside of the cell. S layer and glycocalyx. I don't know why this isn't bolded. I don't know what I did there, but so just, you can bold this if you want. So the S layer is single layers of thousands of copies of a single protein linked together, kind of like a chain link fence. These S layers are produced when bacteria are in hostile environments. So it's a way for them to protect themselves. Glycocalyx also, it is a coating on the cell, repeating polysaccharide or glycoprotein units. And I think there might be a test question about what the glycocalyx is made of. So you might want to make a note of this line here. Examples of the glycocalyx would be the slime layer and a capsule. The slime layer is not the same as an S layer. It is a loose layer and it protects against the loss of water and nutrients. And then the capsule is a more tightly bound, denser and thicker layer on the cell, and it produces a sticky mucoid character in colonies on an agar. So that means when we take this type of bacteria and we introduce it to a particular medium that it likes and it grows, it will look like a mucousy type bacteria. Well, it'll be a colony surrounded by a mucoid layer. So, which is also kind of neat. So if you want to look that up, look up, encapsulated bacteria growing on, blood agar or McConkey or something like that. So you're not going to have to ID these. I'm not going to make you label this, but it's just a picture of kind of how these layers are arranged on some cells. And you can see here's encapsulated bacteria under the microscope. So these are rods, right? And this is all stained with a special stain so we can see these better under the microscope. And, this is the first time you're looking at this. You're not really going to know what you're looking at, but there is like a mucoidy kind of appearance to that as well. I'm trying to think while I'm talking of the one that is most mucoidy that I've seen, but might be Klebsiella. I can't remember. So specialized functions of the glycocalyx, again, to help with capsules. These capsules are formed by pathogenic bacteria or many pathogenic bacteria. They have greater disease-causing abilities, and they protect against, having these capsules protect against the host's immune system, things like white blood cells, specifically phagocytes. So these capsules protect against those phagocytes getting to them. And biofilms are basically armies of bacteria together, and they're much harder to get rid of, but they're commonly found on the plaque on teeth, which protects bacteria from being dislodged. And then also biofilms are responsible for persistent colonization of things like catheters, IUDs, pacemakers that have been implanted, and other implanted medical devices. This is just an illustration of a biofilm and how they're formed. It's actually really interesting, but they have a way of calling out to other cells and kind of just, sending up the alarms, and all of a sudden all these cells, all these different bacterial cells will arrive, and it's much harder to get rid of a biofilm, an infection caused by a biofilm, than it is a single bacteria or single bacterial causing infection. All right, so I'm going to stop there, and we'll pick up on part 3 in the next installment of chapter 3.